Hey Team Bio, okay, sorry for the lateness of the screencast, but this will be a short one, so woohoo. Okay, so last screencast was on the cell cycle, and I might just quickly draw a piece of it out for you here. So if this is the cell cycle, we've got two kind of major phases. There's interphase, which, if you remember from the last screencast, is composed of the G1, the gap or growth phase, then the S phase, which is the DNA synthesis phase, followed by the G2 phase, or the gap growth 2 phase. And then this little red piece of the cycle is mitosis and cytokinesis. Sometimes just called mitosis, but it's also important to remember the cytokinesis or the splitting of the cell piece as well. So we're going to be talking about what happens to the cell, specifically the DNA, at each of these points in both interphase as well as during mitosis, and more specifically about the phases within mitosis itself. Okay, so here's a new blank page, and I'm going to put on an image for you here. Okay, so here's an example of what the DNA looks like in the nucleus during interphase. So, as you recall before, interphase is the majority of the cell's life cycle right here. It spends most of its time in interphase. And usually the DNA kind of looks like this spaghetti type mixture that you can see in here. And this spaghetti is actually chromatin, which you learned about in a reading I had you do, I think before break. Yeah, it was a long time ago. So, as you know, the DNA is a double helix, and it's wrapped around these proteins called histones. And then the histones themselves are coiled up or supercoiled into what's called chromatin fiber. And so this chromatin is kind of like a loose it's like a loose spaghetti, and that allows the DNA to be accessible for both transcription, for transcription and later on translation of that material into protein. So during interphase, the DNA is like this. It's in this loose state in the nuclear membrane. But as the cell moves into mitosis. Oh, and I should also remember interphase is composed of also the S phase, the place where DNA, or the phase where DNA is duplicated. So having DNA in this loose form allows for DNA replication to take place. But as the cell begins to move from interphase into mitosis, the DNA begins to condense into what's called chromosomes. And I'm going to add a picture of that for you here. Okay, so as the cell moves from interphase into mitosis, the DNA begins to condense into a chromosome. And I'm going to break down the pieces of a chromosome for you right here. So it's basically just chromatin supercoiled alongside one another. But there's an important piece of a chromosome. So the chromosome has a couple different, I guess, structures within it. One is called the centromere, which is at the circle right here, and then every chromosome has a couple of arms. You don't really need to know what a telomere is, so I'm going to cross that out for right now. But if you take a chromosome and you cut it down the center of a centromere, then the DNA that is right here up on the chromosome is actually exactly the same as the DNA that's right here. And the DNA that's right here is the same as the DNA that's right here, and so on and so forth. The DNA that's right here is the same as the DNA that's right here. It's because these two things are duplicates of one another. So a condensed chromosome is composed of what's called two sister chromatids. And they are exact replicas of one another. A sister chromatid, or they are formed during DNA replication. So, you might be asking, well, why is it that every cell during mitosis, or actually after the S phase, has two copies of its DNA? Well, it's because that cell is going to split. So it's important that the DNA that is on this side of the chromosome is going to go in one cell, and the DNA that's on this side of the chromosome is going to go into another cell. And so at the end, these two individual cells have the complete, all the genetic material, the whole genetic complement. Okay, so I'm going to throw in another figure for you here. Okay, so here's another figure. It's not the best figure, but it will do. Each, this little yellow, wait. Okay, that was actually a pretty horrible figure, so I found another one. Okay, so here we start out in our figure at G2, gap 2 of interphase. And a few things I want you to notice. Number one, the nuclear membrane here is intact. Number two, the chromatin has been duplicated. So, meaning every chromosome has been duplicated, but the DNA is not in its chromosome form yet. It's its loose, uncompacted stage. And so you can see all the chromatin here looks like spaghetti. And then these centrosomes, they're composed of each a pair of centrioles. This is going to become important later on. You'll see why in a second. Just keep these in the back of your mind. Okay, so we're still in interphase. As we move from interphase into prophase, a couple things begin to happen. Number one, the chromosomes begin to condense into the chromatin begins to condense into its chromosome form. Remember when we looked at the onion root tip cells, you could see this happening in some of your cells. The chromatin beginning to condense into chromosomes. Also, the centrosomes the centrioles begin to migrate to the poles of the cells. These have microtubules coming out of them, which is called the mitotic spindle. This is going to eventually pull the chromosomes apart. You'll see that later on. So that's what happens in prophase. Next we move to a phase which is called pro-metaphase. Some people argue about whether or not this is actually a phase. But in this phase you start to see the nuclear membrane disappear. You also start to see the microtubules attached to the centromere, which is this. There's actually a specialized structure here called the kinetochore. So the spindle apparatus or the microtubules are going to attach to the kinetochores, which is at the centromere, and these chromosomes are going to start to be pulled into the center of the cell. By the time we get to metaphase, all the chromosomes have attachments on both sides of their kinetochores to the centrosomes, so to the spindle apparatus, and they're aligned across what's called the metaphase plate, which is not a real plate, but is just an imaginary line in the center of the cell. So all the chromosomes here, remember, this is the same genetic material as this, and this is the same as this. So eventually when these split, all the genetic material will eventually be two new cells. So they things align across the metaphase plate, the chromosomes align there. Anaphase, they separate from the center, and they are started to be pulled by the spindle apparatus back to the two poles of the cells. What else? Yeah, that's just anaphase. And this happens pretty rapidly. Anaphase is a relatively quick phase. And then the last phase, telophase and cytokinesis, a couple things happen here. The cytoplasm and all the stuff in the cytoplasm that is not depicted here is divided up. So mitochondria end up in both sides. If this is a plant cell, which it's not because it doesn't have a cell wall, then you would see that there would be chloroplasts on both sides. You know, the rough ER has to be divided, the smooth ER, all the different organelles of the cell have to be divided up between these two cells. Additionally, the nuclear membrane starts to reform around what will be the new nucleus, and the DNA starts to unwind from its condensed chromosome state back into its loose chromatin state. So that is all of mitosis. And then again, this is going to be interphase is the next phase after here. And so this is the process by which all your cells grow and divide. So if you have, for example, a cut on your hand, there's going to be mitosis happening in that area, and new skin cells will grow and divide from the existing tissue there. Additionally, when you just grow up, you start as a single cell, and all your cells come from that one original cell that you started as. So all your cells came about through this process, but there are one specific group of cells within you that do not undergo this process, and that is your gamete cells, your eggs and your sperm. Your eggs or your sperm. You're probably not producing both. They go through a special process, and that process is called meiosis. We're going to begin studying it in class on Tuesday. Okay, that is it for today. Yes, that is all. Oh, and get excited because we're dissecting daffodils probably on Monday. Okay, happy Sunday.